Odontogenesis, understanding the science of tooth development. Hello. Imagine you are sectioning a tooth and peeling off its functional hard and soft tissues one by one. The first layer you encounter is the outermost portion of the crown. The enamel, which is the hardest substance in the human body, but is equally brittle. The next layer is the resilient dentin, which forms the bulk of the tooth. Once you remove the first two layers, you can see the pulp, which provides the nutritional and sensory supply to the tooth. The outermost portion of the root is the cementum, surrounding the radicular dentin. The cementum attaches to the alveolar bone with the help of the periodontal ligament. Let us learn how these unique tissues come together to form the remarkable structures that make up our teeth and contribute to our beautiful smiles. Much like the complexity and organization of their structure, the development of teeth, known as odontogenesis, follows a series of meticulously orchestrated steps. We shall study tooth development in detail in a series of 5 videos. This video will be an introduction to odontogenesis. Before we begin, let's test your embryology knowledge with a quick question.
Pop quiz.
Examining embryonic development, we observe the emergence of the stomodeum at around third week of intrauterine life, which represents the initial stage of the formation of the oral cavity. This is formed between the developing brain and the pericardium. Take a closer look at the epithelium lining it, which is otherwise known as the oral ectoderm. It is composed of stratified squamous epithelium duly supported by the connective tissue of ectomesenchymal origin. These unique cells are derived from neural crest cells and migrate to this subepithelial region. These neural crest cells later form the odontoblasts, which form dentin. The oral ectoderm establishes contact with the foregut endoderm, leading to the formation of the buccopharyngeal membrane that blocks the connection between the stomodeum and the developing pharynx. It is on the 27th day of intrauterine life that this membrane ruptures, serving as a pivotal indicator for the commencement of the formation of oral structures, which include the development of teeth. What's fascinating about tooth development is its early onset, around the 6th week of embryonic development, just 2 to 3 weeks after the rupture of the buccopharyngeal membrane. Specific regions of the oral ectoderm exhibit accelerated proliferation compared to adjacent cells, leading to the formation of a horseshoe-shaped thickening called the primary epithelial band. Every individual has two primary epithelial bands, one representing the maxilla and the other representing the mandible. The face is essentially a rapidly multiplying mass of cells. It's important to note that the thickening of these epithelial bands isn't solely due to increased cell proliferation. Rather, it's a consequence of a change in the orientation of the mitotic spindle and cleavage plane during cell division. In the 7th week of embryonic development, the primary epithelial band first divides into an inner or lingual process called the dental lamina, and then into an outer or buccal process called the vestibular lamina. Let's first discuss the dental lamina. Within the dental lamina, 10 localized thickenings or dental placodes develop due to the epithelial outgrowth into the mesenchyme. The maxilla and mandible have 10 thickenings each, representing the 10 deciduous teeth in each arch. This brings us to the next question. Where do permanent teeth develop from? The answer lies in understanding the two unique growth patterns emerging from the primary epithelial band. Firstly, each thickening produces a lingual extension, eventually forming permanent teeth. This is known as the successional lamina. The second growth pattern is the extension of the dental lamina into the distal region, that is, backward, which forms an additional lamina referred to as the accessional lamina. This gives rise to the permanent molars and plays a vital role in guiding their position in the mandibular ramus and the maxillary tuberosity. Note that the permanent molars do not have a deciduous counterpart to develop from a lingual extension. In cases of insufficient distal extension, where the tooth lacks adequate space to grow, complications such as impaction and lingual or buccal eruption of teeth can occur. Now let's discuss the fate of the dental lamina. The activity of the dental lamina spans over at least 5 years. That is roughly the time for the tooth to develop, as we see in the chronology segment of dental anatomy. As teeth develop, they disconnect from the dental lamina, breaking away due to mesenchymal invasion. Some cells persist as islands of odontogenic epithelium, called the cell rests of Serres. These persist as epithelial islands or pearls in the gingiva, above or on the side of the teeth. Cell rests are clinically significant because they can get activated later in life to form cysts, causing delays in tooth eruption, and may even proliferate and differentiate to produce supernumerary teeth. Next, let's discuss the vestibular lamina. It is formed labially or buccally to the dental lamina in both arches. It is also called a lip furrow band, as it hollows and forms the vestibule between the alveolar processes of the jaws and lip or buccal mucosa. Which component initiates and maintains tooth formation? A. Epithelium. B. Ectomesenchyme. The answer is epithelium. This is determined through experiments conducted on mice. The letter E, followed by a number, represents the number of days after embryogenesis. Initially, at E10.5, the epithelium signals the mesenchyme, and these signals are transferred to the ectomesenchyme. However, by E11.5, the ectomesenchymal component takes over and becomes dominant. The epithelial signals involve genes such as LHX and sonic hedgehog or SHH gene. Then epithelial molecules such as fibroblast growth factor 8 or FGF8 and bone morphogenic protein 4 or BMP4. This brings us to the end of our video. In our next video, we will discuss how these dental placodes evolved to give rise to teeth. Hope you had fun learning with us.